Hi everybody, and welcome to another episode of Cell-fie Science. I'm Jen, bringing you a quick burst of science from my cell phone. Have you ever looked at a plant and wondered, how does the water from the roots get all the way to the top of the plant? Well, that's what we're going to talk about today. That process is called transpiration. Now, as you can see in my nice little picture of a soybean seedling here, you've got two systems. What's called the root system, and that's everything that's underground that we usually can't see, and then the shoot system, and that's everything above ground that we can see. When we water plants, water is picked up by the roots. And if you remember when we talked about stomata, gas exchange, including water vapor, happens through the stomata. So water is released from the stomata. That's why they close them when they need to keep water and open up when they can release water. This creates a pressure system, just as it would if you were to blow across the top of a straw with another straw. You get water or soda if you're trying to play a prank on your little brother. Not that I've ever done that. The water goes everywhere. When water is released from the stomata, it has to be replaced. That sets up the pressure so that this water down here in the soil is sucked up, goes up through the plants, replaces in the leaves, and then is eventually released again through transpiration. You can actually measure transpiration pretty simply if you have access to plants and if you have access to clear bags like Ziplocs. I set up two plants 2 days ago. A pea plant and a soybean plant. So these are the peas. If you've never seen peas, this is what pea plants look like. These are about 3 weeks old, and this is some little soybean seedlings. On Friday, I put some clear plastic bags on one set of leaves. So this is one set of trifoliate leaves for the soybeans. And I did the same thing for an upper set of leaves for the pea. And I don't know if you can see here, but at the bottom of this bag, there's some water. That's all the water that was lost from the leaves via transpiration. At the bottom of this bag, there's also some water. To measure this, I'm going to use a graduated cylinder, and we're going to remove the plastic bag from around our leaves. And I taped the bags closed because I knew they might go upside down and I didn't want to lose any water. I'm going to very carefully remove those leaves, see, and they're fine. I'm going to take the water and pour it from our plastic bag into the graduated cylinder. So our soybean plant in 2 days lost almost 4 mils. 3, 4, all right? So that's soybean. Now, let's check out how much our pea plant lost. Again, very carefully, I'm just going to pull this tape off. Remove the bag. Shake off any water that might be on. So this was how many pea leaves were in there. Notice there were more pea leaves than soybean leaves. And we're going to take this and pour it, come on, into our graduated cylinder. Now, notice the pea plants only lost about 1 mil over 2 days. So what does that tell you? If you think that means that soybean transpire more than peas, you're probably correct. Notice the soybean leaves, even though there were only 3 of them, they are much, much bigger than our pea leaves. Greater surface area means more stomata. It also means probably more photosynthesis is occurring because there are more chloroplasts in that area. You can do this in your backyard with any plants. You could look at how does transpiration change with temperature. Maybe you record a oak leaf on a really, really hot summer day, and then you do that exact colder fall day. You might get different results. You could also do what I did and pick 2 different species. These were just soybeans and peas. Perhaps you'd like to try other things that are growing in your garden. Anything is possible, so try it out, and if you get any really cool results, please share them with me in the comments. That's it for today's Cell-fie Science, and I'll see everybody later. Bye!